Thank you. I'm Jamie Brandt. My co-moderator, Sandy Sieminski, and I are looking forward to a great session on delivering solutions. As Dr. Cahook so eloquently outlined in today's Sutro lecture, the paradox confronting us in glaucoma therapy really is that despite having many great treatments, many of our patients fail to use or correctly apply the treatments consistently and thoroughly. In this session, we're going to hear from 6 companies about new approaches to delivering sight-saving treatments to our patients. First up is Randy Craven, who is the Medical Director for U.S. Medical Affairs, Glaucoma Division for Allergan, and he's going to discuss the results of the Zen gel stent trial and the phase 1 and 2 results of a next-generational intracameral implant.
Thank you, Jamie. Also continuing on the theme about what we spoke about today, if I could have my slides up, that'd be great. We see the changes that are occurring for us as pretty significant that is occurring within healthcare right now for all the providers and the patients. And things that are changing, over the years we've certainly seen the importance of IOP become central in what we do with all of our care for patients. And we have different treatment algorithms and different things on the horizon that we can use to help make our decisions better. And the intrusion or the involvement of AI in what's going on for our patient care is becoming evident to us, especially as we meet with so many different key opinion leaders and things that are going on, and decision support tools, as we heard. That's something that we all need help with in making our decisions for taking care of patients. And in the UK, they have an approach where they're really focused on how can we help the patients relieve the burden of what they need to do for taking care of their glaucoma, and can we come up with better methods? So we've heard about the LIGHT trial. Now I would encourage any of you that haven't had a chance to take a look at the Treatment for Advanced Glaucoma study, primarily centered from and driven by Ted Garway-Heath, where they took a look at very specific endpoints at an early phase of what's going on with the differences between medications and trabeculectomy, and there's an indication that it may be more advantageous to have surgery first, and this is different than we'd seen years ago with prior studies. So there's an interest in the UK especially that intervention may be something that we do want to incorporate into our care for glaucoma patients at an early phase. And as we've heard this morning, there's many different options that we have out there available. But one of the things that I think has been underrepresented in our decisions has been the voice of the patient, because so many of our patients struggle with what's going on and what's happening to them, and we're trying to incorporate this in some of our studies. As many of you know, the Zen gel stent is used for helping low IOP in people needing surgical intervention for a filtering procedure. This was a study we did where we took a look at the gel stent versus the trabeculectomy, and we chose an aggregate endpoint which included basically the voice of the patient within this and what happened to them that they perceived as far as their visual recovery, because we felt that it was important for the patient's experience to get put into a part of what happened, similar to how they used the patient's voice in the LIGHT trial a little bit. And out of that, what we did see, if we use that aggregate endpoint, which included how the patient's perception was, it seemed to be that the gel stent performed similar to what we saw with the trabeculectomy with that aggregate endpoint. The other thing that we did see is the differences that were pretty significant between patients' perception of what happened to their eyes after they had a procedure, be it a trabeculectomy or the gel stent, and just what they felt that they had going on and how their visual recovery occurred. So again, the patient's voice is so important. Now, transitioning into where we are right now with our interests at Abbvie especially is with drug delivery. And it's pretty clear as we took a look at and helped with this Kaiser Permanente health record review, and one of our health economic researchers helped with the design of this trial and working with them. But basically, it took a look at over 6,000 patients that had had visual fields done over a period of 5.8 years on the average, and took a look at the slope or the loss of the mean defect in the visual fields and what happened over time. And we did see that definitely those patients that were less adherent on their refill rates compared to those that were more adherent, there was a difference, and there was a significant loss of the visual field over a period of time if you were less adherent. So this is why we think that considering something for drug delivery is key and important for our patients, and trying to work out the right device is a part of what we're searching for. And obviously we want something that's biodegradable, something that can last longer, and something that's as small as possible when we put it into the eye. So our next generation of implants we're looking at now, we have a phase 1-2 dose ranging study that you can find. It's on clinicaltrials.gov, but basically we call it the 408 study, and we've gone through the phase 1 and into the phase 2 and taken a look at parallel assignment compared to that as Lumigan 0.01. Sorry, there's a typo on that there with the dosing. But that's compared with topical Lumigan in the opposite fellow eye. And as we take a look with what we were looking for, the characteristics of the drug that's within the implant, it's high PGA efficacy with some prostamide efficacy as well. And taking a look at the two different doses that we've been looking at and comparing it with the Lumigan 0.01 in the opposite eye, what we see here is on the first dose that we were testing, over the period up to 72 weeks, there seems to be fairly close, as you can see from the graph, representation in IOP between the two cohorts. Small numbers, I will point out with this, but this was our kind of first phase at this, and this is the first time we're sharing this with everyone. Here's the dose B. Taking a look at this, and over the period of time, we're seeing that we're achieving efficacy that we've been quite happy with over the weeks that we've been following these patients. So at Abbvie, we are interested in continuing and working with intervention as an option for taking care of patients, because we think that the burden upon the patients is significant. We're trying to find ways to help reduce that. And we're committed to the research and the development of new things coming along, and we're going to continue to help support that. Thank you.
So we have, I believe, 4 minutes for questions, and we also invite anybody who has a question in the audience to use the microphones here, and there's also QR codes to type in a question. So I'd like to start, actually, as a clinician who's implanting the Zen. What do you see are the future directions with Zen in terms of, are there going to be any new advances or changes to the product, and or how to implant it ab interno versus ab externo?
Yeah, we're committed to continuing to help improve and enhance the success that patients are wanting and needing post-glaucoma surgery. The Zen pipeline, we have several things that are happening globally. The 63 was released outside of the U.S., and we're right now in discussions about looking at that for the United States and a trial in the future, hopefully, but still yet to be determined. But we're also taking a look at what kind of things can individual practitioners do to help improve the success of the patient at the time of surgery. And we've been really quite active in looking at different investigator-initiated trials to help get information on that, educational initiatives to push forward with that. But it really relates to just pre- and post-operative care that we're all, as glaucoma surgeons, familiar we need to do, but helping to share that more with the comprehensive ophthalmologists especially.
Any questions from the audience? Move on.
Do you want me to stay up here then, or?
Yeah.
Okay,
Stay.
thanks.
So when Dr. Brandt and I were paired up to do this session, we got a list of the presenters, and we met in the lobby yesterday, and he said, well, so I'm senior to you, so you take all the difficult names and I'll do the easy names and figure out how to pronounce them tonight. So I'd like to introduce Eyal Shitrit, CEO and co-founder of Eximore. He will discuss the phase 1 clinical trial results for their nanoparticles drug delivery punctal plug. Welcome, thank you.
do I use it? Okay. Good morning. I'm Eyal Shitrit, the co-founder and CEO of Eximore. I'm excited to be here and tell you about our novel drug delivery platform as an alternative to eye drops to deliver a solution to various eye diseases. As you all know, and Malik also gave us a lot of explanation about the challenges of the ophthalmic drug delivery today. Patients do not adhere to the necessary medications, other common problems, overuse, low drug bioavailability, and side effects. Eximore was founded to solve this problem and to create a viable alternative to the eye drop market. At Eximore, we have developed a unique and proprietary pack-and-release punctal plugs. Here are the three essential properties of our innovation: high drug capacity, sustained release drug delivery, and plug retention for extended treatment period. How I go back this way? Okay. The plug utilizes the patient's natural tears to release the drug into the surface of the eye through nanochannels that provide a slow, sustained release of the drug according to the treatment dose. Although a microplug is tiny, our innovation is in using nanoparticles that offer a vast surface area and high drug capacity. 1 gram of these materials has a surface area of 600 square meters. Our plug is placed inside the tear ducts in the doctor's office in a safe, simple, and easy procedure. Depending upon the treatment plan, the plug, an undegradable polymer, is replaced every 3 to 6 months. Based on our unique design and technology, we have been able to differentiate Eximore from both eye drops and other drug delivery systems. Our clear value proposition suggests a better way to deliver drugs, eliminate patients' compliance, and use no preservatives. Compared to other drug delivery systems that have been tested and developed over the past several years, we offer the following: a minimally invasive procedure, an extended treatment period, the capability to pack more than one drug, and the option to treat in multiple indications, as our portfolio shows. With a focus firstly on glaucoma, with the prostaglandin latanoprost, and secondarily on dry eye with tacrolimus. So let's dive into our clinical and preclinical data. We completed both in vitro and in vivo studies which showed in vitro controllable drug release at the optimal therapeutic rate and plug biocompatibility tests with no findings of concern. In the in vivo studies, Eximore's glaucoma product demonstrated an impressive 50% pressure reduction in the canine model. Having all this in place, we proceeded to phase 1 clinical trials in both glaucoma and dry eye. We recently completed clinical studies for both indications with more than 40 patients. Our results indicated good safety, tolerability, and excellent efficacy. This supports our product as a potential alternative solution to the challenges of eye drops that require lifelong medical therapy. Let me share more details about our clinical studies and results. In the glaucoma study, we compared our plug to xalatan eye drops as a control. After a washout period, we measured the pressure baseline, inserted the plug, and followed up for 3 months. The study focused on safety and efficacy. First, for safety, the plugs showed no serious adverse events. Almost all adverse events were mild and expected, with a similar number of adverse events reported in the control eyes. Second, the plugs were well tolerated and showed a 24% pressure reduction from the baseline. Here is the efficacy data of our study in a greater detail. As you can see from the graph, 24% pressure reduction on average for the 3-month study. The plug was non-inferior to the eye drop control treatment and performed better compared to data from other punctal plugs. In addition to phase 1 study in glaucoma, we completed also a phase 1 trial in the dry eye. We compared our plug to a commercial silicone plug as a control. Similar to the glaucoma study, the results showed no serious adverse events. The plug were well tolerated and received significantly better scores than the commercial silicone plugs. With this very encouraging data from phase 1 studies, we look forward to advancing our clinical program in the US and abroad. The key components of our plan for the next 2 years include the following: completing phase 2 US study in glaucoma, continuing preclinical work in other indications, expanding the team, and lastly, securing the necessary capital to advance this plan. To conclude, Eximore's nano-based technology is novel, safe, and effective at packing and releasing APIs to treat multiple ophthalmic indications. Our results are thanks to our experienced team, including our scientific, medical, and business advisors. Thank you for your attention, and I welcome the opportunity to interact with you during the conference.
Thank you very much. This paper is open, or this presentation is open for questions, and you're welcome to step up to the microphone. Let me get started. One of the challenges in punctal delivery has been retention, because we all worry that we put something in, and if a patient comes back 6 months later, it's not there. When did it fall out? Do you have retention data in your clinical trials?
Yes. We actually put a lot of efforts on these issues because we are familiar with the competitors and other solutions in the market. I think we solved this problem in terms of 2 months with more than 90% retention rate, but for the 3 months we are still enhancing our results. We are trying to add more ideas how to increase and improve this retention rate. We also would like to use a mobile app to monitor the use of or to locate the punctum in its place. And this is another idea how to make sure that our patient is treated well.
I have a question, actually. So I noticed in your the dry eye study, you were looking at a lot of corneal measures like TBUT and OSDI. Did you look at them in the glaucoma study? Because I would think there might be a difference just having a punctal
Yes,
plug.
that was part of the one. Yeah.
So what did you find?
For what?
For the latanoprost study, looking at the corneal surface.
We didn't see any big changes during this period. It was more of the same. I mean, it was stable.
Any questions from the audience?
Okay, there's a question here that you, I think, already addressed. Did the plugs fall out for any of the patients in the phase 1 study? There was a question here from the audience. No, there was a question here from the audience.
From you.
Did the plugs fall out for any patients in the phase 1 study?
Sorry,
Here's the question from the audience. Did any patients in the phase 1 study lose the
No, well, most of them kept the plug in place for the rest of the studies, but some of them they had a fell out of the plug, and that was because they touch in this area, and then we had to re-instruct them how to make sure not to touch this.
Okay.
Yes.
Great. Thank you.
Thank you.
Thank you.
Thank you very much.
So it's my pleasure to introduce Tomas Navratil, PhD, who is the Chief Development Officer at Glaukos, and he'll be discussing Glaukos' drug delivery device, the iDose, along with the iStent Infinity and a new drug delivery platform we're all eager to hear about.
Thank you so much, Jamie, and thank you to you and Sandy for the opportunity to present. I'd also like to congratulate Adrian, Andy, Michelle, Tom, and the team behind you. This has been another terrific Glaucoma 360 meeting, and it has really become my favorite glaucoma meeting of the year. So with that, it's my great privilege to provide you an update on the progress achieved within the Glaukos R&D pipeline, as it has been a terrific year since the last time we saw each other at the last Glaucoma 360. We will, of course, focus on the glaucoma pipeline in further detail. These are our disclaimers. Here, I'd like to acknowledge that this work would not be able to proceed without contributions from many investigators, collaborators, colleagues from the FDA, some of whom are in the audience, so thank you so much for all that you've contributed. We could not do what we do without you. So more than 20 years ago, Glaukos was founded based on the basic premise and intent to transform glaucoma therapy, which originally led to the iStent and creation of new medical device class, the topical bypass medical devices. But as you know, our ambitions and aspirations go much further than that. And so one key thought I would like to leave you with here today that has been implemented at Glaukos since its inception has been to challenge and disrupt the traditional paradigm of topical IOP-lowering eye drop use and eye drop use in general. So based on this basic intent and principle, we have invested over $400 million in R&D over the last 5 years. And with this investment, we have been able to advance multiple therapeutic platforms, each of which is capable of spawning multiple products that challenge the traditional paradigm. Here you can see some of the platforms, the iStent trabecular bypass device platform that has been greatly extended, the iDose intracameral delivery system, the iLink bioactivated pharmaceuticals, iLution transdermal dropless cream delivery through the eyelid, and finally our Retina XR utilizing biodegradable retinal implants. Based on these platforms, we have been able to generate a formidable pipeline, including 7 marketed products available across 17 markets worldwide, and about 14 R&D pipeline products, all together targeting multiple diseases that affect more than half of the patients, half of this planet's population. So today, I would like to share with you 3 vignettes focusing on progress within our glaucoma R&D pipeline. The first vignette focuses and highlights the iStent Infinite product, which has been cleared in the second half of last year. It is important to note here that this product was studied in difficult-to-treat glaucoma patient populations. Patients came to this trial based on baseline 3.1 IOP-lowering medications and 2 prior failed incisional surgeries. Despite this baseline background disease, we have demonstrated 6 millimeter change from baseline at month 12. We have also showed high percentage of responders, with 76% of patients demonstrating and meeting the 20% or greater change in IOP from baseline, and more than 50% of patients experienced 30% or greater treatment effect. So it is important to note that this was the first standalone approval of the trabecular bypass device that now can be administered apart from cataract surgery. We are, of course, expanding this indication and are intent to studying milder and moderate glaucoma patients in the next step. The second vignette I would like to share with you today is our iLution Travoprost transdermal dropless eyelid delivery cream. The iLution platform has been created really to offer easy administration, potential faster onset of action, and fewer side effects due to the reduction of the exposure of the surface to the preservatives. So with the iLution Travoprost platform, we have now submitted to US FDA IND at the tail end of last year, and we are intent on starting first time in human clinical trial near term. So we are very excited about this program. Please stay tuned. And lastly, the third vignette and the final vignette I'd like to share with you is focused on iDose. iDose really was designed fundamentally to address the ubiquitous patient non-adherence and the chronic side effects that we see from topical IOP-lowering medications. iDose is a miniature intracameral implant that is anchored at the iridocorneal angle and is capable of delivering multiple years of IOP-lowering pharmacotherapy to glaucoma patients. In the completed phase 3 program, we are pleased to share with you that iDose met primary efficacy endpoint in both trials, demonstrating IOP lowering ranging from 6.6 to 8.5 millimeters of mercury. Please note that in this trial, we have studied over 1,100 patients, more than a quarter of which came to the study on 2 or more IOP-lowering medications. And despite this pretty heavy background pharmacotherapy, we have seen that at the completion of 12 months of evaluation in both phase 3 studies, we had 93% responder rates for patients who remain well-controlled for their IOP on the same or lesser number of IOP-lowering medications compared to baseline. Additionally, we had 81% of patients at 12 months, based on both phase 3 studies, who remain completely IOP-lowering medication-free. These results are strongly corroborated and based on the completed 3-year evaluation in a phase 2b program, wherein at the completion of 3 years, we still had 69% of iDose patients who were well controlled for their IOP on the same or lesser number of topical IOP-lowering medications. And this 69% rate favorably compares with the 45% of patients who were well controlled in the timolol control arm. So please note here that if you look at the single iDose, compared to 2,190 eye drops that the patient had to administer in a timolol control arm over the course of 3 years, we may offer patients benefit in reduction of the pharmacotherapy burden. The safety and tolerability has been evaluated both short-term and long-term in the course of 36-month evaluation in the completed phase 2b program and in the course of 12-month evaluation in the phase 3 program. It is notable that there are no adverse events of periorbital fat atrophy, very low to no conjunctival hyperemia, no adverse events of corneal endothelial cell loss in these 12 and 3-year evaluation, 12-month and 3-year evaluations, and very low or no incidence of iris color change. So in addition to these completed phase 2 and phase 3 trials, we have conducted the so-called exchange study that was designed to allow readministration of iDose. In this study, we have studied second implantation cycle in eyes that were previously implanted with iDose for an average period of 5.3 years. Over this period, we have seen no incidence of adverse events of corneal endothelial cell loss and no decrease in the overall corneal endothelial cell counts, which is quite an exciting outcome. So in summary, iDose delivered and demonstrated favorable long-term efficacy and safety. Additionally, the exchange trial, which will be submitted as part of this imminent NDA filing to allow readministration of iDose, provides an exciting outcome for our patients. And lastly, it's notable that number of sites who participated in the phase 3 study actually delivered iDose as an in-office procedure without changing outcomes that were favorable for the overall phase 3 program. So in summation, the Glaukos therapeutic platforms have led to generation of this formidable glaucoma pipeline that targets all stages of the glaucoma disease progression. So in summary, we truly believe that Glaukos is different, as we are delivering multiple disrupting dropless technologies that have multiple advantages over topical eye drops. And given the recent progress with iDose, iLution Travoprost, iStent Infinite, and other pipeline products that include PreserFlow, iAccess, iPrime, and second and third generation products based on the iDose technology, we truly believe we carry forward a high degree of momentum and are truly executing on our core mission, which is to improve lives of glaucoma patients worldwide. Thank you so much.
All right, Tomas. I told you I would ask you a challenging question.
Thank you, Sandra.
Here it goes. So with the ATR exchange, what were any adverse events that you saw in your trial in terms of just the actual exchange? So were there any reports of endophthalmitis or any reports where you could not retrieve the original one?
Thank you so much for the question. Both of these areas were some of the areas that we studied very closely. So in the course of the exchange, the way the exchange is conducted is new iDose implant is inserted, and then without removing the inserter from the anterior segment, the old iDose container is removed. In this study, we had 100% success of removing the iDose and exchanging, and we also saw no incidence in endophthalmitis. So we feel really satisfied and really excited about the outcome, and are submitting this trial to allow readministration of the iDose with the original NDA submission.
Thank you.
Okay. Any other questions from the audience? Text? Okay. Thank you.
Thank you so much.
Okay, so you're introducing.
So I'd like to introduce to the stage Therain Tu, CEO and co-founder of Qlaris Bio. He will discuss the clinical results of the Qlaris 111, a refined formulation of a potassium channel modulating medication, which selectively targets episcleral venous pressure.
It's just a clicker here. Hello, everyone. I'm Therain Tu, CEO and co-founder of Qlaris Bio. It's definitely a pleasure to be presenting to you today. Just to give you a quick overview of what we are doing at Qlaris, Qlaris is focused on right now developing a novel platform of ATP-sensitive potassium channel modulators that are serving as a complementary mechanism of action to other therapies and interventions available on the market right now that are going to be targeting selectively episcleral venous pressure. With that selective mechanism of action, we are hoping to provide products that will be able to lower pressure in patients with hypertensive patients, as well as normotensive patients by delivering clinically meaningful IOP reductions while also providing an excellent safety and tolerability profile with no hyperemia. But before I continue, I would like to, on behalf of my co-founder, Dr. Barbara Roscoe, our investors, and the entire Qlaris team, thank the organizers for having us present on Qlaris Bio here, and especially Dr. Andrew Iwa and Adrian Graves for allowing us to present today. So one might ask, why are we developing further eye drops for glaucoma? When you think about how many available medications are on the marketplace right now, the market does seem quite saturated. However, if you look at these medications, they are all targeting 3 out of 4 components of IOP currently, predominantly aqueous humor formation, uveoscleral outflow, and conventional outflow facility. None of these therapies currently target selectively episcleral venous pressure. And by not targeting episcleral venous pressure, you're actually leaving the largest component of IOP untouched and unaddressed at this point in time. So at Qlaris, we are targeting especially episcleral venous pressure using ATP-sensitive potassium channel modulators as a vasodilatory mechanism that can reduce EVP and also improve distal outflow resistance. Our initial formulation, QLS 101, was based on research by Professor Michael Fausch at Mayo Clinic. It is that formulation that we had originally rapidly progressed into the clinic, into our first in-human studies. In those studies, we were actually able to demonstrate a very excellent safety and tolerability profile that avoided hyperemia over the entire 28-day period. Moreover, in those studies, we were also able to substantiate IOP-lowering efficacy that gave us confidence to move forward with this platform of ATP-sensitive potassium channel modulators. Now, I'm actually happy to present data with our latest formulation of this platform called QLS 101, which is a refined formulation that also happens to remain preservative-free. So now, I'm happy to, again, state that we have been able to, with our collaborators, execute upon an investigator-initiated clinical study in 21 normotensive patients. In these patients, they were offered either a low or high dose of the new QLS 111 formulation or vehicle to compare and see whether or not we have a remaining excellent safety and tolerability profile, but also significant IOP lowering. What we were able to find, and I'm mentioning the data right now, is that with QLS 111, we were able to lower, after 14 days, intraocular pressure in excess of 20% reduction in mean diurnal IOP. That equates to a 3.5 millimeter reduction from a baseline of about 16.7 millimeters of mercury in these patients, but also importantly, maintaining that excellent safety and tolerability profile where we saw absolutely no reports of instillation pain, burning, or stinging, no ocular findings whatsoever, and most importantly, no clinically significant hyperemia in any of these patients. So what might this do for us? By targeting EVP, we believe now we have the potential to treat a range of patient populations for whom EVP had been to this time limiting in either pharmacotherapy or has in itself been pathologic for patients. So this includes diseases such as normal tension glaucoma, where currently patients are limited by the floor that is set by EVP. This is a large patient population, as you can imagine. It is also disproportionately affecting Asian patients as well. And in normal tension glaucoma, there are currently no on-label approved therapies. This goes beyond, again, primary open-angle glaucoma, which we're very familiar with, that this program may be very complementary and additive in terms of adjunctive use. But also, this includes therapies such as interventional surgical therapies like MIGS devices that actually shunt flow, or that is aqueous humor, into the episcleral veins, and we believe we'd be a perfect complement there as well. But this also does include diseases where EVP itself is pathologic. That includes pediatric rare diseases like Sturge-Weber syndrome, where EVP is significantly elevated. This therapy may be able to much more specifically and selectively target the pathology in these patients as well. So certainly what we might consider is that QLS 111 has the ability to really provide substantial value and significant potential in the marketplace across a number of different and disparate patient populations with different forms of glaucoma. So in summary, we'd just like to close by saying with QLS 111, we've had the opportunity now to establish proof of concept for the novel mechanism of action that is now targeting episcleral venous pressure by leveraging ATP-sensitive potassium channels. What we hope with this program is to now bring meaningful IOP reductions in patients where this has been limiting, and this includes, as we mentioned, large patient populations like normal tension glaucoma. This mechanism as well, again, is complementary to other mechanisms that are out there right now, given the uniqueness and specificity of its mechanism. And we are now very confident that we have a highly de-risked program that we are going to be moving forward with into phase 2 studies later this year in both primary open-angle glaucoma and ocular hypertensives, but also in normal tension glaucoma patients going forward. We're excited to be able to present this opportunity once again, and we are now going to be raising an additional $25 million Series B for Qlaris. So we look forward to providing everybody further updates as we go along, and we're certainly excited to bring forth to you the value of targeting episcleral venous pressure. Thank you.
So as somebody who spends a lot of my time dealing with pediatric glaucoma, I have a large population of Sturge-Weber patients. Do you have any data at all or any experience in patients with Sturge-Weber?
Yes, right. No, that's a great question, Jamie. And what we have right now is actually we've been starting to, with our original formulation, assessing the safety and tolerability of this mechanism in Sturge-Weber patients. We have not publicly shared this data yet, but we do have data in terms of the ability to target these patients, and it is a category that we are looking forward to developing in the future as a broader aspect of where we're bringing value to patients.
Do you have any other questions?
Anybody in the audience? I just had a question on systemic.
There's somebody coming up, but
Okay.
go ahead to the microphone.
I had a question about systemic side effects
Yes, ma'am.
quickly. Did you see any as a vasodilator in terms of blood pressure, anything systemically?
Oh, that is a great question. So as you know, this category has been evaluated for its antihypertensive effects systemically. At the doses and the level of exposure we are providing from an ophthalmic drop at the doses that we are providing, we are not seeing anything that is of significant concern whatsoever.
Thank you.
Just a real quick question.
Yes.
I'm James Burke from Vices.
Hi, James.
Did I miss this, but is the lower dose more effective than the higher dose?
That is a great question. Again, we just want to be very clear and give a caveat, right? These are relatively small studies, and in these small studies, it is very hard to determine whether or not any discrepancies in terms of actual efficacy are meaningful. What we do believe, however, is that we are providing at these doses an already adequate level of exposure to the drug, to the target channels. And because of that, we may be actually already exceeding what might be efficacious and hence maybe not seeing any further efficacy. But it's hard to really determine based on these small numbers whether or not there is a significant difference between the two doses just yet.
All right. So thank you very much.
Great. Thank you.
Join us. Oh, there. Our next speaker is Thomas Reeves, who's the co-founder and president and CEO of Ripple Therapeutics. He's going to discuss the 6-month preclinical data for RTC1119 which is an intracameral latanoprost prodrug, as well as RTC 620, an intravitreal bimatoprost implant.
Thank you very much. Appreciate the opportunity of speaking. And I think I've figured out that I am the solution to California's drought, because every time I'm in town, it rains and pours. So I'll make sure I visit regularly, but not too frequently. So very pleased to be here. I think Dr. Cook described very well the issues with current drug therapies. Obviously, eye drops work well, but compliance is an issue, and ocular side effects complicate the issues. Surgery is, by definition, invasive. And polymer-based implants work, but they've had little uptake because of the issues with corneal endothelial cell loss with multiple treatments. So Ripple's approach is very different. We've discovered a way of creating processable prodrugs that allow for very high drug loading, smaller implants, and these are 100% the prodrug, the implant here that you see on the left. And the drug release is based on surface mediation. So it's constant daily drug release, and it degrades over time. So by the time the implant is gone, the drug is gone. It's very predictable and very tunable based on either the linker chemistry or the physical dimensions of the device. And because of this simple monomeric structure, it's very scalable and low cost. So it's a platform technology, but we'd focused on ophthalmology because what we see is the opportunities in multiple diseases where the current therapies have challenges, whether it be cost, compliance, efficacy, or duration. So we have a full pipeline of both retina and glaucoma products. The FDA has given us guidance that the 505(b)(2) path is appropriate for our lead programs on both retina and glaucoma. We have composition of matter patents for a number of our drug conjugates, and we have commercial traction with our lead retina program, having licensed this to TEA for North America and Europe and CSP Pharma for China. So I'm going to talk about our glaucoma program, but just wanted to share with you the clinical data from our dexamethasone prodrug that's treating DME and RVO, because I think it's evidence of the translation of this technology to the clinic. So the target product profile here is 6 to 12-month drug release with a smaller implant that degrades fully over time. We're delivering 1/10 of the drug of the leading commercial product, which we believe will lead to lower incidence of IOP and cataract AEs. You can see here on the left, your really nice preclinical PK and PD data, which translate to that target product profile. And on the right, we've just finished enrollment, 60 patients in a phase 2 trial in Hong Kong, Australia, New Zealand, and Canada. And you can see here just the nice reduction in CST, and you can't see it on these slides, but the actual improvement in vision for patients in both DME and RVO, both naive and treated patients. And so again, kind of clinical validation of this technology. Our glaucoma pipeline includes both front of the eye applications for IOP lowering and back of the eye applications for IOP lowering and neuroprotection. And I'm going to focus today on our lead program, RTC 1119, which is a latanoprost prodrug implant, intracameral implant. So again, target product profile is controlled drug release for 6 to 12 months with a small needle and 100% prostaglandin prodrug that doesn't swell and doesn't have any inflammatory degradation products. And again, the important thing, no polymers or excipients, so when the implant is gone, the drug is gone and there's retreatment potential. I think if you look at the right there, it kind of describes well our implant shrinks over time, whereas polymer-based systems, and the Drist is an example here, swell over time. If you look at this graphic, I think it really describes why we believe that we won't have the same issues. So with traditional polymer-based systems, once the drug is gone from the initial implant, you still have the polymer carcass there, and so the second implant can potentially stack and touch the corneal endothelium. When the polymer degrades, you also get pro-inflammatory acidic degradation products, which can also cause damage to the endothelium. Whereas ours is smaller, it doesn't swell, so it sits lower in the iridocorneal angle. Once the drug is gone, the implant is gone, and so you can add that second implant and not have the same issues, either from a physical or a chemical point of view. We've got a real battery of preclinical data. This is our rabbit study, which I think shows a good example of the degradation of the implant over time and how it settles really nicely in the iridocorneal angle. Good safety profile, including no histological findings at either 4 months or 8 months. Our dog study, again, very solid safety profile, both the ocular exams and the pachymetry. And in the PD study, in normal tensive dogs, you can see there that We are in line with Durista's IOP lowering at 90 days, and ours continues out, in this case, to 9 to 10 months. And then the pupillometry data, I think, is a good example. The fact that with the Durista product, the drug is gone at 90 days, where you can see that our drug continues to work in terms of pupil restriction, again, out to that 9 to 10 months. Right now, we're focused on developing the preclinical and CMC package to start a phase 2 trial, 20 to 30 patients with topical latanoprost in the fellow eye. We've also focused, as mentioned, on an intravitreal product. This is what we call RTC 620, which is a bimatoprost intravitreal product. Rabbit tolerability and safety data looks good. Just this last week, we started our PD data in canines, so look forward to those results in the next few months. We've made a lot of progress on both of these products, and we think that there's a number of different milestones over the next few months that'll be value inflection points. We're in the process of raising a Series B financing. The primary use of funds is to focus on taking those 2 primary glaucoma programs through the non-clinical CMC and phase 2 clinical. So look forward to meeting investors or strategic partners interested in learning more about Ripple. Thank you.
I had a question. You mentioned in your canine study looking at pachymetry. Did you look at corneal endothelial cell count?
We did, but because, so we looked at it and there was no issue, but we don't have multiple implants, so it's really hard to draw a conclusion from that the multiple implants would not have any corneal endothelial cell loss. But we think because of the mechanism of action of shrinking over time and being gone when the drug is gone, that we don't have the same issues that others have had.
Thank you.
Other questions from the audience? Please don't be shy. Somebody's coming up.
I have a question about why you would choose intravitreal for one glaucoma platform and intracameral for another. Was there a reason for that?
Sure. So when we looked first at the intracameral, we felt that the latanoprost had the right product profile in terms of the time of release. And for intravitreal, we think we need a stronger drug, a more potent drug to be able to get the drug to the target tissue. So we've looked at both for both, but those are the 2 that we think are the best combination.
All right. So this is a generic question for anybody with implant studies. Did you have aqueous humor taps, and do you see changes in biochemistry of aqueous humor over time?
We did do the taps. We didn't see any change in that humor. I mean, we can measure the drug in the aqueous humor from the intracameral, but we didn't see any changes other than seeing that the drug is there.
So you don't think there is a more permanent change to the aqueous humor or the ciliary body that's measurable at least?
Do you mean could it last longer than when the drug is actually gone?
Yeah.
Potentially, but we didn't really study that in the canine or the rabbit models.
We have one audience question. Have you looked at placing your implant in the subconjunctival space?
Short answer is we thought about it, but we haven't done it.
Okay.
Okay.
Great.
Thank
Thank
you.
you. So it's my pleasure to introduce. Oh, you're next. Go for it.
You can do it.
Oh, Peter, come on up. Peter Solstig is the Global Head Product Development Division and the Chief Medical Officer for Santen, and he's going to discuss Omlonti, which is a new prostanoid EP2 receptor agonist, which was launched in Japan in 2018 and can be useful in patients who are non-responders to FP agonists.
Thanks, Jamie. Good morning, everyone. I'm glad to hear my name is seemingly not that difficult since Jamie could pronounce it. So good morning again. I'm just going to give you a quick update with regards to Omlonti. Omlonti is a purple for glaucoma. This was approved end of last year, so just a quick update for you. So Omlonti is a purple, is a topical prodrug. This is hydrolyzed in the eye during corneal penetration to omidinipag, which is a selective non-prostaglandin prostanoid EP2 receptor agonist. And if you look at it from the left-hand side, you can see the changes when omidinipag isopropyl becomes hydrolyzed to omidinipag, and also how this compares from a chemical structure also versus typical FP agonists such as that of latanoprost. Omlonti has a novel mechanism of action. It is recognized as a different ATC class by the WHO. So if you look on the right-hand side, you can see where it is located, so to speak, in the nomenclature. It is together with drugs such as natazidil, ripasudil, versus the typical prostaglandin analogs of latanoprost, travoprost, and so forth. The mechanism of action, as I mentioned, targeting EP2 receptor, but it has a different pathway in the sense that it actually activates both the conventional as well as the uvoscleral pathway. So what does Omlonti try to do? Well, with its novel dual mechanism of action, favorable safety profile, omidinipag may be a useful IOP lowering agent. For instance, in patients who have shown themselves to be, so to speak, non or low responders to PGA such as FP agonists. We have conducted a variety of clinical studies, to be specific, 12 as of today. OMDI, or omidinipag isopropyl, has been available in Japan since 2018, as well as in Asia now for quite some time. Now, in interest of time, I'm just going to be focusing upon three main studies for today. First one is the Peony trial, which was conducted in Asia, India, Korea, Singapore, and Taiwan to be specific. This was a trial that compared omidinipag versus latanoprost, approximately 200 patients for 3 months. Then Spectrum 4, which was conducted here in the US, and I'm sure we have many investigators here with us as well, so thank you for those who participated. This trial compared omidinipag versus timolol, again, approximately 200 patients for 3 months. And then finally, Spectrum 5, which looked at latanoprost low non-responders, also here in the US, approximately 100 patients for 3 months. Let me share some data. This is from the Peony trial, where omidinipag achieved comparable efficacy to latanoprost. You can see here that this is the phase 3 randomized trial versus latanoprost, and you can see that it had a comparable IOP reduction between omidinipag versus latanoprost. Spectrum 4 trial, if we're looking here, this is the trial versus timolol. Again, we were looking at the primary endpoint, which was non-inferiority versus timolol. And you can see that the difference in mean IOP was within the 1.5 milligram mercury at all 9 time points, both when you're looking at weeks 1, 6, as well as 3 months. Another way of looking at the data from Spectrum 4 is, of course, you're looking at the responder rates of mean diurnal. These were comparable. Again, if you look at 20% reduction, 25, 30%, as well as patients, the percentage of patients who were responders defined as achieving less than 80 milligram mercury response rates, and highlighted in orange with OMDI, you can see here that it was comparable, so there were no significant group differences in the response rates weeks 1, 6, and month 3. If we look at Spectrum 5, this is data from the Spectrum 5, which was a open-label, multi-center phase 3 trial, which was also conducted here in the US. It was studied in a bit more than 100 patients. Patients were first put on latanoprost, and patients who basically didn't have a IOP reduction after 8-week run-in period, defined as less than a 15% change in the IOP reductions, had then the opportunity to continue them with Omlonti. And what we could see here is that we achieved an additional 3 milligram mercury reduction with Omlonti. However, again, would like to just caution that these results were not powered to show statistical significance and should be interpreted with some caution due to its open label design and the lack of comparator. From a safety perspective, Omlonti provides a predictable and well-tolerated profile. Most common adverse events you can see here was conjunctival hyperemia, which was approximately 9%. The events were mild and non-discontinued due to conjunctival hyperemia. And you can see here the overall percentage of patients who discontinued due to AEs. So to sum it up, omidinib isopropyl, we believe provides a novel, a well-tolerated option to effectively lower IOP. We believe that unmet need still persists in terms of IOP control, as we've heard already, with regards to tolerability, as well as a side effect profile. This is a novel EP2 receptor agonist, achieves efficacious and sustained IOP reduction via activating multiple pathways, and has a predictable and well-tolerated profiles without any major concerns in terms of also of adherence and surgical outcomes. That concludes my presentation. Thank you very much.
Yeah. I had a question about it. I know it's a prostanoid receptor agonist rather than a prostaglandin. Do we still need to avoid patients who have active CME, active uveitis, in practice in Japan and so forth? Are they practitioners?
Short answer, I would say yes.
Yes.
Yeah.
Are there other questions from the audience? Can you talk a little bit further, other pipeline approaches in the Santen and the glaucoma space? I know you have more than just this one drug. Just an overview.
Yes. Well, we don't have the presentation here, so I'll just briefly mention it.
Sure.
I mean, Santen is a global company, so we have the presence, obviously, in several countries, and depending upon the unmet needs in those countries, we also have different approaches. But particularly here in the US, we most recently conducted our trial for sapatoprost, or 126, which is a FP3 receptor agonist. That study, the phase 2 trial, compared sapatoprost versus timolol. We could also show there the non-inferiority. So that is, I would say, our next phase 3 trial, which is planned to be ongoing. That trial is actually already in stage 3 in Japan. And then, of course, we have in Europe, we have also a cachoprost, which is a cationic emulsion of latanoprost. So that one we have also submitted in Europe. So really, as a global company, we have a different pipeline. So, but
Great.
yeah, that's in a brief summary.
Great, thank you. We were wondering, how are we doing on time? Do we have a minute or two?
We need to wrap it up.
We need to wrap it up? Okay.
We're going to get taken off the stage now. Thank you to the panelists. Thank you so much for participating in this great discussion. I also want to give a special shout out to Michelle, because she really did a lot of background work looking into all these companies and helping us decide.